There's a child buried in a cave on Mount Carmel in Israel, a 5-year-old. The skeleton was discovered in 1932 and sat in museum archives for decades, classified as an early Homo sapiens and largely forgotten. Then in 2025, researchers from Tel Aviv University and the French National Center for Scientific Research put it under advanced micro-CT scanners and looked again, really looked for the first time. What they found stopped them cold. The fossil is the earliest known human fossil in the world to display morphological features of both Homo sapiens and Neanderthals. The skull vault curved like ours. The jaw and inner ear were Neanderthal. This was not an ancient human. This was not a Neanderthal. This was the world's first known human hybrid, the earliest solid physical evidence of interbreeding between Homo sapiens and Neanderthals, pushing back the timeline of human interbreeding by more than 100,000 years. A 5-year-old child, 140,000 years old. Two species that scientists had been calling separate, coiled together in a single small skeleton in a cave in what is now northern Israel. And here's what that means. Everything you were taught about the boundary between human and non-human, every clean, confident line drawn between our species and the beings that came before us, rests on a foundation that is now actively collapsing under the weight of new evidence. Not because scientists were fraudulent, because science works, because every decade, new tools reveal what older tools could not see. And what they are revealing, consistently and from multiple directions simultaneously, is that the line between human and ape does not exist the way we thought it did. It may not exist at all. To understand why this matters so much, you have to understand what the old story looked like, and how completely it dominated everything. For most of the 20th century, human evolution was taught as a ladder. At the bottom, apes. At the top, modern humans. A clean, progressive march from primitive to sophisticated, from animal to something categorically higher. Every classroom had the same image, a series of increasingly upright figures, each slightly smarter and more refined than the last, culminating in a modern man striding confidently into the future. It was a comforting image. It placed us at the apex of a long, inevitable ascent. It made us feel like the point of the whole enterprise. That image was wrong. Scientists knew it was a simplification. But simplifications, repeated long enough, become dogma. And this one shaped not just biology textbooks, it shaped philosophy, theology, law, ethics, and the entire framework through which Western civilization decided what counted as a person and what didn't. The latter is gone now. What replaced it is stranger, messier, and far harder to explain. Instead of a straight march from ape-like ancestors to modern humans, researchers now see a tangled, branching tree with multiple species coexisting. At various points in our prehistory, there were not one, not two, but at least 6 other human species alive when Homo sapiens first emerged in Africa. 6 different kinds of people, different skulls, different bodies, different tools, different continents, and they all disappeared. Every single one of them, except us. That is the context that is almost always missing when people talk about evolution. We are not the destination of a forward march. We are the sole survivors of a much larger family. And understanding what we are exactly in relation to those who didn't make it, that is where all the certainties begin to dissolve. Begin with genetics, because the genetics is where the story becomes genuinely explosive. For decades, school children were told that humans and chimpanzees share 98 to 99% of their DNA. This fact became one of the most repeated statistics in the history of science. It was quoted by the Smithsonian Museum of Natural History, by Bill Nye, the science guy, and in major scientific journals like National Geographic, Science, Scientific American, and even Nature. The American Museum of Natural History in New York put it on their website. Humans and chimps share a surprising 98.8% of their DNA. There was one problem. The number was produced using incomplete genome data, and the methodology had a flaw baked into its foundation. Most previous genome-wide comparative studies of apes had been limited by the mapping of inferior assemblies to a higher quality human genome. Consequently, human reference biases were introduced. Scientists were measuring chimp DNA against human DNA by using human DNA as the reference framework. The ape genomes were being assessed through the lens of our own genome, which made them look more like us than they actually were. It was like measuring how foreign a language is by using English as the benchmark, and then concluding that everything resembles English. Then in April 2025, a landmark study published in Nature changed everything. Scientists sequenced complete ape genomes from scratch without using the human genome as a guide, using advanced long-read sequencing technology that had never before been applied at this scale. The results were stunning. Sections of human DNA without a clear counterpart in chimp DNA make up approximately 15 to 20% of the genome. Some bits of DNA are present in one species but missing entirely in the other, known as insertions and deletions. When comparisons include harder to align regions, that difference pushes closer to 5 to 10%, and if we account for regions still too complex to align with current technology, the true overall difference likely exceeds 10%. The actual difference between human and chimp DNA is 14 times greater than the often quoted 1% statistic. 14 times. A number printed in every textbook, repeated in every classroom, cited in thousands of peer-reviewed papers, was off by a factor of 14. Not because anyone was lying, but because the tools weren't good enough, and the methodology introduced a systematic bias that nobody corrected for until now. This does not mean humans and chimps are unrelated. They are deeply related. But the story of exactly how close, and what that closeness means, is far more complicated than the clean, confident number suggested. And the complications are where the real science lives. Here is where it gets philosophically destabilizing. The genetic differences between us and chimps lie mostly not in the genes that build bodies, but in the regions that control how genes switch on and off. The differences between human and chimp genomes lie mostly in non-coding DNA, SPAS, the segments that do not code for a specific protein and that make up about 98% of the genome. While coding DNA contains the instructions for protein building, regulatory regions found in non-coding DNA control how, when, and where proteins are made. They act like switches, controlling whether a gene is turned on or off. This is critical. It means the biological difference between a chimpanzee and a human being is not primarily a difference in ingredients. It's a difference in the recipe, the timing, the sequence, the order in which developmental programs activate and deactivate. Two bodies built from almost the same parts, assembled according to different instructions, producing one creature that lives in trees and another that builds spacecraft. And one of the most consequential differences lies in a single gene that didn't even exist in our last common ancestor with chimps. ARGAP11B arose from partial duplication of ARGAP11A on the human lineage after separation from the chimpanzee lineage. It is, in the most literal sense, a human-specific gene, one of the molecular signatures that makes us anatomically distinct. The human neocortex is roughly 3 times bigger than that of our closest relatives, the chimpanzees, and is the seat of many of the higher cognitive functions that are unique to humans, such as our speech or the ability to learn. ARGAP11B is one of the key drivers of that expansion. When scientists inserted it into developing primate brains in controlled experiments, the neocortex grew larger, began to fold, and produced more of the upper layer neurons associated with higher cognitive function. A single gene. A single copying error that survived and spread because it produced a brain that could do things no previous brain could do. But here is what that also means. There is no place in the genome where you can point and say, here, this is where the human begins and the ape ends. The boundary doesn't exist as a bright line. What it looks like, examined honestly, is a gradient, a set of differences that accumulate across a genome so vast and so complex that scientists are only now beginning to understand what most of it even does. And if the genetic line is blurry, the fossil record destroys it entirely. While many believe Homo sapiens evolved in East Africa around 200,000 years ago, groundbreaking discoveries suggest a different story. In 2017, fossils found at Jebel Irhoud in Morocco, on the northern coast of Africa, were dated to over 300,000 years ago and are now believed to be the earliest known remains of Homo sapiens, pushing back the timeline of human evolution further than previously thought. Those Moroccan skulls had modern human faces, flat, with small teeth, recognizable features, but the backs of their skulls were long and low, shaped more like Neanderthals. They looked like us from the front and something else from behind, which raises a question paleoanthropologists are still actively debating. Were these fully human? A transitional form? Or does the question itself reveal the problem with the categories we're using? Newly discovered teeth in Ethiopia reveal a previously unknown species of Australopithecus that lived alongside some of the earliest Homo specimens nearly 2.8 million years ago. This suggests that nature tested multiple versions of being human before our lineage endured. Nature tested multiple versions of being human. That sentence deserves to be held in the mind. Evolution was not building toward us. Evolution was running experiments, and we happened to be the experiment that didn't go extinct. At least not yet. Fossils unearthed in Ethiopia are reshaping our view of human evolution. Instead of a straight march from ape-like ancestors to modern humans, researchers now see a tangled branching tree. Lineages that dead-ended, species that flourished for hundreds of thousands of years, and then vanished entirely. Species that coexisted, competed with each other, and, as the evidence now makes undeniable, did something else with each other entirely. Now we come to the part that most people have not fully absorbed. And it is time to absorb it. We did not just live alongside Neanderthals, we had children with them. Those children survived. Their descendants are alive and reading these words. Modern humans and Neanderthals didn't just cross paths, They repeatedly interbred, shared genes, and even merged populations over nearly 250,000 years. These revelations suggest that Neanderthals never truly disappeared. They were absorbed. A Princeton University study published in Science documented this process across 2 full centuries of millennia. Not a one-time encounter on the edge of a migration route. Not a single isolated event. A sustained, repeated exchange of genetic material across an almost incomprehensible stretch of time. People of non-African descent derive 1 to 4% of their genomes from this extinct human cousin. And then, in August 2025, the child in the cave on Mount Carmel pushed that story back further still. The fossil from Skull Cave establishes the Levant as the cradle of humanity's first mixed population, and the research team behind the study confirmed. This discovery reveals the world's earliest known human fossil, showing morphological traits of both of these human groups, which until recently were considered 2 separate species. That phrase is worth reading again. 2 species that science textbooks treated as categorically distinct. They had a child together 140,000 years ago. That child was buried. Someone put that body in the ground with intention. Whatever we want to call the people who did that, they were not purely one thing or another. They existed in the space between our categories. And it wasn't only Neanderthals. People of East Asian descent not only have a greater percentage of Neanderthal genes, but also genes, up to 0.1% of their genome, from another early hominin group, the Denisovans. A group whose entire existence was unknown to science until 2010, when a fragment of finger bone from a cave in Siberia was sequenced and returned a genome unlike anything in the database. Not Neanderthal, not sapiens, something else, something that had been living alongside us, interbreeding with populations in East Asia and across Oceania, leaving genetic traces in millions of living people today. We did not find them with a skeleton or a skull. We found them inside us. Some of the Neanderthal genes involved in the immune system and skin pigmentation actually increased in frequency in Homo sapiens over time, implying that they may have been advantageous to human survival. We didn't just breed with our evolutionary cousins. We borrowed their adaptations, their cold weather immune responses, their metabolic physiology. The environment changes, and then some genes become beneficial. Neanderthal genes, dormant for tens of thousands of years, became critical survival tools when new diseases arrived, when climates shifted, when our populations pushed into environments they had never occupied before. We are, in a very literal biological sense, part Neanderthal, part Denisovan. The boundary between us and them is not a wall, it is a membrane, and it has been permeable for longer than civilization has existed. Now, return to the language question. Because if anything was supposed to mark the definitive boundary between human and non-human, it was language, the capacity for complex, recursive, symbolic speech. The ability to talk about things that aren't present, to discuss the past and the future, to tell stories, to lie. For years, scientists pointed to a gene called FOXP2, known in the literature as the language gene, as evidence of human uniqueness. Mutations in FOXP2 produce severe speech and language impairments in humans. The gene was found to have 2 human-specific changes relative to chimps. This was presented as a molecular signature of what makes us singular. Then came the Neanderthal genomes. Researchers found that our closest extinct relatives, the Neanderthals, share with modern humans 2 evolutionary changes in FOXP2, the gene that has been implicated in the development of speech and language. The results suggest that these genetic changes predate the common ancestor of modern humans and Neanderthal populations, which existed about 300,000 to 400,000 years ago. The gene we identified as uniquely human, the one we used as molecular proof of our singular capacity for language, Neanderthals had it too. The entire framework collapsed. Researchers analyzing genetic data from a diverse sample of modern people and Neanderthals found no evidence for recent human-specific selection of FOXP2, fundamentally revising the history of how we think humans acquired language. And in songbirds, birds whose vocal learning is structurally analogous to human speech. FOXP2 is essential for learning songs, offering a striking parallel to human language learning. The gene we called the language gene is present in functional form in birds. The boundary we drew using molecular biology dissolved just as completely as the one drawn using fossils. Consider what all of this, taken together, actually means. There's a species called Homo naledi, found in the Rising Star cave system in South Africa. A small-brained hominin dating to between 335,000 and 241,000 years ago, found deep within a cave system accessible only through a narrow, nearly vertical chute. Preliminary evidence from the excavation suggested these beings may have carried their dead deep into the cave deliberately, may have made engravings on the cave walls, and may have used fire to navigate the darkness. The claims remain contested. Peer review has raised serious questions about the evidence, and the scientific community is divided. But the fact that the debate is happening at all, that scientists are seriously arguing about whether a small-brained hominin with a brain a third the size of ours may have performed ritualistic behaviors, tells you everything about where the science currently stands. We cannot draw the line confidently even there. And we know with far greater certainty that Neanderthals buried their dead. We know they cared for their injured. A disabled Neanderthal found in a cave in Iraq, known as Ned, had only one arm and one working eye, yet was aided and kept alive by his Neanderthal community, showing evidence for compassion among the species, seemingly in contradiction from any behavior based solely on evolutionary advantage. Compassion in Neanderthals. The species we spent decades portraying as brutish dead ends of evolution kept their disabled alive. Whatever cognitive and emotional capacity must be present before a group of beings chooses to feed and protect a member who cannot hunt or defend himself. Neanderthals had it, and their genes are in us now. Whatever Ned was, part of him never went anywhere. Paleoanthropologist Ella Al-Shamahi, who presented the landmark BBC and PBS series Human in 2025, and whose career began, improbably, as a Muslim creationist trying to disprove evolution at university, puts it in terms that cut through the scientific complexity and land in the gut. Scientists once believed they could pinpoint the moment when humans first appeared. But as new fossils have filled in our evolutionary history, that defining moment has begun to disappear, replaced by a far stranger picture of what it means to be human. A far stranger picture. That is the key phrase. Not a more depressing picture, not a more reductive picture. A stranger one. One in which the beings we shared this planet with for hundreds of thousands of years were not simply lesser versions of us on the way to becoming us, but parallel experiments, equally ancient, in some ways equally complex, some of whose DNA we carry in our bodies to this day, and which we are only now beginning to understand. Everyone from outside sub-Saharan Africa, and even some people within sub-Saharan Africa, have some Neanderthal DNA in them. And we were also interbreeding with quite a few of those other human species that existed alongside us. We were interbreeding with quite a few of them. Multiple species. Multiple times, across hundreds of thousands of years, on multiple continents. And the biological evidence of those encounters is written in living human genomes, in the immune systems of people who have never heard the word Neanderthal, in the metabolic pathways of populations across Asia and Oceania, in a 5-year-old child buried in a cave in Israel that confused everyone who looked at it for 90 years, until 2025, when the technology finally caught up with what the bones had always been trying to say. So where does the line actually fall? Scientists are quite sharply divided over who or what our human ancestors even were. The fossil record is full of gaps, not because transitional forms didn't exist, but because fossilization is extraordinarily rare. And we have only been searching a tiny fraction of the timeline behind us. In 2026, an international team identified a previously unknown fossil ape from 17 to 18 million years ago in Egypt, the first definitive evidence of an ape from North Africa, reshaping the entire story of where our evolutionary lineage began. Every new discovery doesn't just add a data point, it moves the goalposts. It erases what seemed like a firm boundary and replaces it with a question, and the questions keep getting more fundamental. What makes us human? Is it walking upright? Neanderthals walked upright. Is it tool use? Chimps use tools. Crows use tools. Octopuses, under certain conditions, use tools. Is it language? Orangutans communicate using a complex repertoire of consonant-like calls that researchers compared as consonant-based speech. Is it compassion? The Neanderthal with one arm and one eye refutes that claim. Is it art? We have found ochre pigment and perforated shells from over 100,000 years ago, likely created by beings we do not fully classify as modern humans. Is it abstract thought? We have no idea what Neanderthals were thinking. We only know they buried their dead. We only know they kept their disabled alive. We only know they had the FOXP2 gene, the gene we call the language gene, identical to ours. Is it the size of the brain? Homo naledi had a brain a third the size of ours and may have carried its dead through a narrow underground passage in the dark, using fire to see. The scientists arguing about whether that happened are arguing about it because the possibility cannot be ruled out on brain-size grounds alone. What then? What is the line? The honest answer, the one the evidence is pointing at from every direction simultaneously, is that the line is not a line. It never was. What it is a gradient so gradual, a transition so slow across millions of years, that on any given day, in any given generation, nothing changed enough to notice. And then one day, through an accumulation of changes so small that no single one of them would have qualified as a threshold crossing, something was capable of looking at the sky and wondering what was up there, not because survival required it, but because the brain that had been slowly, expensively, metabolically, radically enlarged over millions of years by a gene called RGAP11B could not help it. That wondering, that specific, strange, costly habit of asking questions that have no immediate survival value, may be the only trait that comes close to separating us from everything that came before. And even there, we cannot be certain that Neanderthals did not wonder too. We cannot hear them thinking. We can only see what they left behind. A 5-year-old child in a cave in Israel, a man with one arm kept alive by his community in Iraq, ochre-stained shells strung on a cord, bodies carried by torchlight through underground passages that no one can reach without squeezing through a crack barely wide enough to pass. These are not the traces of animals, but they are also not exactly us. They are something in between, something that our categories cannot hold, something the science keeps finding as it digs deeper and sequences further and dates more precisely, something that refuses to disappear no matter how hard the old story tries to keep it contained. We certainly weren't exceptional in the beginning, but now we're the only ones left. That sentence should sit with you. We were not exceptional. There were others. There were multiple experiments running in parallel, some of them nearly as complex as us, some of them intertwined with us genetically, some of them burying their children and keeping their injured alive and possibly making marks on cave walls in the dark. And every single one of them is gone. Everyone, except the descendants of the ones who absorbed them, except us, who carry their genes and their immunities and their adaptations in our bodies, who are in the most literal biological sense a composite of everything that came before. The line between human and ape doesn't exist. What exists is an unbroken chain of life, billions of years long, that produced every organism on this planet, including the ones reading these words. The question of where on that chain we stand is less important now than the question of what we do with the position we occupy. Because we are the only ones left. We are the only version of this experiment still running, and we are only now beginning to understand what that actually means.